Folks. So I said something yesterday in a video that I made on microbial ecology. There will be a test number 4 on that material, but I'll post that on the 17th. I'm going to wait until you're done with this virus test, and the due date on it is the 16th. I think at 11:59 p.m. So make sure that you get in there and get that done. Of course, put in some time reviewing the material before you do that. Some of you have already taken it and scores are looking really good, so that's encouraging. Um, so there's that. So yeah, wait on that test for. Um, in the meantime, what I'm going to do is put some stuff up relating to lab, okay? And I'm going to give um, a couple of abbreviated sort of exams over this material. It's the best way I know how to evaluate student understanding. I'd rather be doing this stuff hands-on in the lab and do as we normally would do. Of course, that's not possible. So there are actually three things. I said a couple of three things I'm going to be working with you on. Selective and differential media. That was one of the labs we would have done. A lab on something called MVIC testing, which is done to test for gram-negative coliform organisms. That's an important thing to be able to do. Um, and then a final coverage of several organisms, bacteria that you should know. And so after each of those, I'm going to put up a short assessment of 15 or so questions, and your performance on those is what I'm going to use to assess your lab grade. And so um, I'm barred from going on campus to get anything, and I had a bunch of lab grades in Excel there. So until someone gives me the go ahead, this is how I'm going to have to do it. All right, so I think it'll work out. I'm not going to make these crazy hard or anything like that, but standard meat and potatoes type of questions. So let's get started. The first one of these from lab that I'm going to go, and by the way, you have a lab eLearn shell. We just don't use it much in lab because I cover everything on the board and just go with that. But these are up there, and you can access them. So this one is on bacterial identification and the use of differential and selective media. Let me go ahead and pull it up. We'll start this slideshow. All right, and so you can see it goes back a little ways, but the information hasn't changed, so we still use this. Okay, so one of the things that was discussed earlier in lecture actually was the fact that in an investigation where you're trying ultimately to identify an organism, you have several steps that you go through. We talk about the 5 I's of microbiology: incubation, inoculation, isolation, information gathering, and then finally you arrive at identification if it's something that has shown up in sampling of the environment or sampling of food or water, and we don't know what it is. We had better try to figure out what it is so that we know how to deal with it, to know whether it's a potential pathogen and that sort of thing. So identification is a major goal, and there are multiple ways that we can go about doing that. So there's that figure from a previous chapter in Talaro, chapter 3. We've already covered that and tested on that. But one way to go if you're trying to get to where you can identify organisms, if you want to isolate them from mixed communities, if you've got a bunch of different species occurring together, and you want to isolate particular ones, you can use streak plating, which is where you take an auger plate and apply a sample, and then in a serial fashion, with flaming the loop in between um, uh, streaking serial quadrants, you ultimately spread the organisms out to the extent that you'll see individual colonies, and you can pick which ones you want to work with and culture them separately, and there you have it, pure cultures. You can do that. Another way to go is to use what are called selective media. All right, so selective media actually do select particular forms out of what potentially might be a mixture of many, and allows you then to focus on particular groups based on their metabolism and other features that they have. So the definition here, it should be emphasized that these media contain ingredients that inhibit the growth of many species and allow others to grow. All right, so basically many of them nutrient augers or nutrient broths, to which we've added additional things to modify them to make them specialized for particular jobs. And so those things can include dye chemicals, and you might ask, why would you put a dye in a medium? What's that going to do? Well, dyes penetrate the envelope of an organism. They penetrate the cell wall and perhaps even the cell membrane. So certain organisms are especially sensitive to that. And so a dye might inhibit them. A dye might obstruct, passage of important nutrients across the cell wall and cell membrane of a particular organism. So dyes can be helpful sometimes in inhibiting some organisms and allowing others to grow. Of course, antibiotics, which again, don't kill viruses. Somebody's been saying that in the media recently. But antibiotics kill bacteria. They don't work on viruses. I'm sorry, azithromycin is not going to kill viruses. But yeah, you can include an antibiotic, and it turns out that some antibiotics are especially effective against the gram positives. And so if you include an antibiotic that is especially potent on, as far as killing gram positives, you will select gram negatives, and those are the ones that'll grow. So right away, you winnow down the possible number of organisms to a smaller ensemble of things, and that's very helpful typically. And then you can include, or exclude, as the case may be, various nutrients, and those two can help to favor the growth of some organisms while inhibiting others. So you can use selective media. And again, the object here is to take what potentially is a confusing large number of species and to, basically jettison a bunch of them, get rid of a bunch of them, and leave behind a few that you can work with that you're really interested in. That's the whole goal. Of course, now we also have differential media. And so if you have a medium that is strictly differential, it's not going to reduce the number of organisms that you have in a mixed culture. But what it will do instead is highlight the differences between them. I know we've talked about this before. This is kind of a lecture thing, but it bears repeating. And it is something that we deal with in the second half of the lab. So if you weren't fully clear on what differential media were previously, now you should be. These are media that, let's say you have 15 different organisms in a mixed culture, and you plate them out on the differential medium. You'll still grow 15 organisms, but you'll see major differences in them that are brought out by constituents, that is, ingredients of the media that you're using. So that's generally what you're going to expect with them. So they're differential. all right, because they contain ingredients that enhance differences, all right? Differential, difference, get it? Those two go hand in hand. That's why we call them differential. They differentiate. That is, they highlight the differences among organisms. And then, of course, there are media that accomplish both of those things. And generally speaking, that's most of the media we would work with. Just, you know, honestly, that's typically how it works out. If a medium is selective, it's also differential. But you know, we have a few that fall clearly into one category or the other. Sometimes it's worth reviewing that. You have a bunch of different media, kind of categorizing them on the basis of which of these groups they fall into. Strictly selective, strictly differential, or are they both? Those are the three possibilities. So the first medium I'm going to talk about, and these are very widely used, important media, all of them that we're going to talk about. We're not going to go through a bunch of them, some major ones. So if you were starting a microbiology lab, for sure you're going to have nutrient agar and nutrient broth, and then you would probably want to lay in a supply of this type of agar, EMB agar. And this stands for eosin methylene blue. So the E is for eosin and MB stands for methylene blue. Those are both, of course, dyes. You've worked with methylene blue before and doing simple staining. And then we also have eosin, which actually has a more kind of a maroon color, I think. But both of those are dyes that impregnate and pass through and combine with the thick peptidoglycan of a gram-positive. Right? So they will ultimately inhibit gram-positive metabolism, and gram-positive organisms will not grow well on this type of medium. So this is a medium that selects for gram-negatives. Right? And frequently gram-negatives are pathogens, serious pathogens, and so if there's concern about the presence of gram-negatives in a particular setting, in water, in food, the environment, where people might be active, using this type of medium would determine if they're there and might even help you figure out what kind they are, because not only is it selective, but it is differential. Right? It differentiates as well. And part of the reason it differentiates is that it has lactose in it, lactose sugar. This is an important carbon source for some organisms. Some species have the ability to ferment lactose and get energy from it. Others don't. They would have to use other sugars. So that's genetically based, by the way, that difference. Things that can ferment lactose have genes for that, and as a consequence, enzymes for that. Those that can't ferment lactose don't have those genes. Okay. So there are constituents in this EMB agar that would give you a specific colony color that would indicate whether or not lactose is being fermented. Generally speaking, the organisms that ferment lactose will produce really strong, really dark-looking colonies on this agar. Right? And the agar itself is dark, but you can see that the colonies are dark. They have kind of a deep purple look to them. Now, if it's a really, really strong fermentation response, it goes beyond that, and you'll see a metallic green sheen on the surface of the colonies. So that's really strong lactose fermentation. So the results with this are, no lactose fermentation would be where the colonies are clear. A moderate amount, a decent amount of lactose fermentation, blue-black colonies. said purple, but more blue-black. Now, if it's really good, outstanding lactose fermentation, and there's some organisms that do that, and not a bunch of them, so that's helpful, right? If you were to see it, you would know, probably um, a few possibilities. You'd have narrowed the range of possibilities down quite a bit, and so this is where you see the metallic green look to the colonies. It's on the surface of the colony. So this plate that's shown here, you can see it's got a bunch of metallic green looking colonies on it. All right. It actually was quadrant streaked plate, streak plated. It was done that way, and in every one of these quadrants, you've got these organisms here. So it's pretty much a pure culture of whatever this one organism is, and it turns out that it's likely to be E. coli. E. coli is one of the main ones that does this. And there are a few other forms that do it. There's an organism called Citrobacter, and we used to use this a lot. And haven't, and well, in a year or so we haven't used it, but there's different species of Citrobacter. I'm not going to write out the species name because it confuse you, but it's Freundii. Anyway, that's EMB, and it's really useful, again, for selecting and differentiating gram-negative organisms. The reason that gram-negatives, as I said, are significant is that many of them are pathogens, and there are also a number of them that are found in fecal material. So if they are present, say, in a lake, you got a place like Beach Lake, and people are going there swimming, and somebody goes and samples the water, and they find gram-negative organisms in there, especially those that can ferment lactose really strongly on this agar, then you probably have some fecal contamination. That's what that indicates. And so sewage is getting in there, and obviously that's not a good thing. So that's one sort of scenario where an EMB agar would come in very handy, is in testing for that sort of thing. And here's another plate that shows some EMB agar and how organisms are responding. So this metallic green sheen here, you see how this is an organism that was sort of streaked on there in a fishtail fashion. That's more than likely E. coli. And then this organism here is some organism that's fermenting lactose. So we would say that it's lactose positive is usually how that's designated. That means it ferments lactose. Whatever that organism is, it's fermenting lactose, but not to the same extent as E. coli. Right? It's doing a good job at fermenting the lactose, but not quite like E. coli or, say, Citrobacter or something like that. And then over here, you can see there's growth. You can tell that there's something there. But if you look at the color of the growth, it's clear because you can see the agar on the other side of it. Right? So these are clear colonies. This is an organism that's probably a gram-negative, but it is lactose negative. Right? Got lactose negative, a lactose positive, and then E. coli. We could designate that maybe with 2 pluses because it's really very strong lactose fermenter, or whatever that organism is. It could be some other one of those. All right, does that make sense? So that's EMB, eosin methylene blue agar. Another agar type, I probably should have covered this one last, but because there's another agar type similar to EMB that would be a good one to follow up with. But anyway, we'll talk about this one. So this is mannitol salt agar, typically, obviously, referred to using an acronym, as is the case frequently with things that have long names. So this is MSA, mannitol salt agar. And Obviously, some of the constituents include mannitol and salt. The salt is just sodium chloride, a certain percentage of that. I think it's like 2% or something, which means 2 grams of sodium chloride for every 100 milliliters of agar um, uh, suspension. The mannitol is it is a sugar. It's one of those what are called sugar alcohols. Things there's things like sorbitol and xylitol. These are commonly used in you know, um, diet kinds of foods and things like gum that's advertised as being sugar free. They don't have sugars like fructose and glucose and sucrose, but they have these sugar alcohols sometimes. Um, and they can be good carbon sources for organisms. Some of them are equipped with the enzymes and, of course, then the genes to be able to handle mannitol and use it as a carbon source. Well, some of them do that. Others don't. Right. So if you've got mannitol in there, and some organisms are plated on that agar and they can't use mannitol, well, they're not going to grow well. All right. So this is going to select for mannitol fermenting species. Right. And the salt, in the recent lecture on microbial ecology that I went through, that salt, when it's present in the surroundings, increases the solute concentration of the environment the organisms are in and tends to have a desiccating effect. It actually pulls water out of cells, right? Osmosis causes water to go toward the salt, essentially, into the hypertonic surrounding conditions. So some organisms can resist that really well. And so again, salt is in there as an agent to help select for particular organisms. Got those two things together. So there's another thing that's in this agar too. It's a pH indicator that gives a red color, and it's due to the presence of this substance known as phenol red. Phenol red is a pH indicator, and in acidic conditions, it turns yellow. All right. And what would cause acidic conditions? Well, it's fermentation of mannitol. So if you see the red changing to yellow as organisms grow on this stuff, it indicates that there was mannitol fermentation. So that could be helpful too. Now, there are some organisms that can grow on this that won't use the mannitol. Right? They'll be fine to grow on there because they can utilize the other nutrients that are present. Others may not be able to. Okay, so you can have, here's the possibilities: no growth at all. That would indicate that the MSA has inhibited organisms entirely. Growth without yellow color means an organism can get by on this stuff and do well. It obtains nutrients from other sources, and it's not affected by salt. Right? It's a salt-loving organism. And then the other possibility is the organisms grow and they use mannitol and produce acid, and you get a color change. Okay? So you can have no growth, you can have growth without color change, and you can have growth with color change. Those are all three possibilities. Now, it turns out the kinds of organisms that grow well on this agar are limited to only those that are halophilic, salt lovers, and generally those are going to be found on your skin. Why? When you sweat, you leave behind some salty residue there. Your skin is always a little bit salty, and that's actually going to be inhibitory. It can prevent the entry of certain species that might possibly be pathogens. So that's a helpful thing. Now, there are organisms that are adapted to it. right? So you have a skin flora, and among those are the Staphylococci. And you can see here the two common species are Staph aureus and Staph epidermidis, right? Aureus is the one that potentially is pathogenic. Uh, Often it isn't, but there are strains of it that can be. You've heard about MRSA and VRSA. Those are strains of Staph aureus that are antibiotic resistant, methicillin resistant, and vancomycin resistant Staph aureus. So if you are sampling some, you know, somebody's skin or some other surface, and you get organisms growing on MSA that ferment the mannitol and produce a yellow color, they could potentially be one of those strains. Yeah. More than likely they aren't, but that's at least you know the possibility exists. Epidermidis, Staph epidermidis, doesn't ferment mannitol. So you get good growth because it's a halophile, but it's not going to ferment the mannitol. You're not going to see yellow color. And then another genus that's common in the skin floras of different folks is Micrococcus, and it contains several species, and many of those are members of the skin flora, so they'll grow on this auger. They're halophilic. So something like a gram-negative organism like E. coli, it isn't going to grow on this. The vast majority of the coliforms won't grow on this because they're not adapted to salty environments. Staphylococci, the micrococci, and some few others actually will grow well on this. So here's a mannitol salt auger plate, and you can see it's divided in half, and on one side they streaked an organism that's growing there, but it's not fermenting mannitol. Here we've got one that's growing and it's fermenting the mannitol. You can see the yellow color starting to diffuse out into the surroundings. So this side is probably Staph epidermidis. Of course, that's more than likely Staph aureus. So there's that. Okay. Mannitol salt auger. This is a good one for investigating gram-positive cocci. Gram-positive cocci are among those that are commonly found in the skin floras of organisms. And so they'll grow on this, and we can differentiate them on the basis of mannitol fermentation. All right, this is the one I said probably should have been covered immediately after reviewing EMB because this is more closely related to what EMB does. This is McConkey auger. In our lab, we usually designate it as MAC to indicate we got a plate of this and it's sitting alongside something that looks similar to it, and there's a few things that do look similar to it. We'll usually designate it with an MAC to clarify what it is. You see, it's kind of like MSA auger in appearance. It's not dark sort of purple, blue, black like EMB, but has more of a light red color to it. Not quite as bright red as an MSA plate. Got a little bit more of a subdued look to it. Just a tinge of purple in there. But anyway, this is McConkey auger, and it's used for studying gram-negative forms that live in the gut. Another term for those is enterobacteria. And I wouldn't have said anything at all about that if it wasn't for the fact that it's here. So that word needs explanation. Entero refers to the gut. Okay? Your gut, in the old Latin, I guess it is called the enteron. You've heard of gastroenteritis and dysentery and all of those things. That ENTER is referring to your your gut, your alimentary canal. And so these um, organisms that are found there in the gut flora are primarily gram negatives. Not all of them are. If you want to cultivate those, there are some media that are especially useful for that, and McConkey is one of them. And it's unique in that it has a bile salt. Okay, so bile, of course, is released into the duodenum by way of the bile duct, um, along with pancreatic juice. Every time some chyme from your stomach passes into the small intestine, so bile is going to be integrated into that material that's passing through the gut. And it turns out it's got properties that tend to be harsh and inhibiting to a lot of organisms. Okay? So the reason the gut flora is different from the flora, say, of your skin is because many of the organisms that are there are there because they can handle this bile, this onslaught of the bile that's coming through. I think it's hypertonic stuff, and it has kind of a emulsifying action, which tends to break down cell membranes and that sort of thing. So anyway, there's bile in it. It inhibits some organisms and doesn't inhibit others. If something doesn't live in the gut, if it's not an Enterobacteriaceae, one of those forms, it probably is not going to be able to handle that bile. So bile is the inhibitor, right? It's included in here as an inhibitor. And then there's lactose. Okay, so that's something that it has in common with EMB. And then it's got this substance known as neutral red, okay, different from some of the dyes that are found in other media. This neutral red is one that will bind with the end products of lactose fermentation. So after lactose is fermented, there are some products of that. And neutral red tends to bind with those and create dark colors. So darker, say, purples and pinks, as opposed to you see this kind of neutral red color that this has. All right, so you see any kind of red, any kind of dark pink, purple, darker colors like that in colonies that are growing on McConkey agar, that tells us that it's a lactose fermenter. If you have growth and you don't see that those darker colors, it's just creamy white or clear, then it's not a lactose fermenter, right? So among the things that live in your gut that don't ferment lactose, but which are especially important for us to be able to identify, are members of the genus Salmonella, right? And I don't know what dysentery group means because there's a bunch of organisms that can cause dysentery, some of which aren't even bacteria, but this one here in particular, Salmonella, is a genus that contains some species that, as you know, are pretty serious potential pathogens. And so these species, most of them will grow on McConkey agar and EMB agar even, but they're not lactose fermenters. Okay? So that can be helpful. E. coli is a lactose fermenter. We know that from our discussion with EMB. Well, it would do the same on McConkey. All right. So here's McConkey agar. And we've got growth here. Now you can see E. coli, again, is strongly lactose positive. And this is like hot, sort of fuchsia colored almost, right? Really, I guess that's fuchsia or magenta. I don't know. I don't know all the specific colors, but you can see that's really a strong response. And it's not limited to just the growth, which is in this streak here. You can see it kind of going out into the surroundings too. Okay? But what that must mean is this organism is producing an enzyme that it's secreting out of itself into the surroundings, and it's fermenting the lactose out there. And enzymes that are like that get secreted into the surroundings, are called exoenzymes. So there are a number of enzymes that could be at work there, lactose dehydrogenases and things like that that organism might have that it's using to digest that stuff. And then here we have another organism, and it's got this nice pink color, purple, um, and but it's limited to the growth. And so that's a positive response, but it's not to the same extent as E. coli. So we've got some forms like that. And of course, you can see there's growth here, right? But the color behind that plate is, but behind the growth in the plated agar underneath, that's what you're seeing. You're looking right through the growth at that agar, so it's clear. And so the organism can grow on this, but it's not fermenting the lactose. There's other things it's using to get by, and it's able to withstand, that is, it can overcome the inhibiting effects of the bile salt. This might be salmonella, all right? Might be salmonella that's growing in that situation. So there's McConkey agar, and I can't really tell you why the two different agars are out there. They're both commonly used. I don't know if there's some people that prefer EMB for, say, testing environmental samples, and McConkey is used for testing body fluids and that sort of thing. I'm not sure. They both give similar results, and it may just come down to personal preference. But you can see it's plainly obvious when you have a lactose fermenter, that really stands out with this agar. Again, gram negatives. This is going to favor the growth of gram negatives, many of which are gut flora members, and so it selects for those, and then it differentiates them on the basis of their lactose fermenting abilities. Here's another look at McConkey agar. And you can see, here's a salmonella species. This is Enterica, right? So there's the Entero root word right there in the species name of this particular salmonella species. And you can see there's no dark color in those colonies. So salmonella forms are liable to do that, whereas in the center of each one of these colonies of E. coli, you got a nice dark, in this case, red color. That would tell us that it's a lactose fermenter. There's that. So that's the end of that. All right. So I'll be posting here shortly a little quiz covering some of that stuff, but quiz, exam, sort of in between. It's a little longer than a typical quiz, but it's shorter than a typical exam. I'm going to use that as one of the things to assess your performance in the lab. So be looking for that. All right, so I'm going to stop that there. And I'll be putting up another video shortly. So watch your eLearn and watch for that to come across.